Hi, my name is Tony Tien.
My name is Pilar Martin, and today we will be discussing the characterization of nanosensors sent to us by the Prud'homme Research Group.
So the Prud'homme Research Group at Princeton sent us nanosensors that were made with flash nanoprecipitation, also known as FNP. And FNP is a technique used to make nanoparticles that utilizes rapid mixing. And it's particularly touted for its ability to load a lot of active agents into nanoparticles, which can be useful for drug delivery. However, we're using them for oxygen sensing.
We tested 10 different nanosensor samples that had varying compositions of polystyrene PEG, Di-A, PTTPP, and vitamin E or vitamin E acetate. In these sensors, PTTPP acts as the oxygen responsive dye, and Di-A acts as a reference dye, which is used as a baseline for comparisons.
So to find the best sample out of the 10 samples to use, we used a glucose-glucose oxidase test to simulate a deoxygenated environment in comparison to ambient oxygen. And we used the fluorescence data from that to determine which of the samples would be the best. And so in looking at the oxygenated to deoxygenated signal ratios, we found sample 5 to be the best after then evaluating which samples had the highest fluorescence overall. So it beats out samples 3, 4, and 6 due to higher resolution. And the data we used to look at that with involved the peaks for Di-A, which is the reference dye that doesn't change much, as well as PTTPP, which is the dye that changes in response to oxygen.
We tested the oxygen response of sample 5 using a laser and a gas bubbling system. Figure 6 shows that PTTPP responds to varying oxygen concentrations as expected. Di-A also responds to different oxygen concentrations, which is unexpected as the optimal Di-A peak would look more like figure 5, where there is little variation between samples. Figure 7 shows that the nanosensors are reversible as we were able to cycle back and forth between 0% and 21% oxygen without affecting response.
So in conclusion, FNP is a promising platform for improving the efficiency and scalability of making nanosensors, particularly for oxygen. It seems like it's a promising solution, though with our particular formulation with Di as our reference dye, since it varies with oxygen concentration, we don't have a good baseline, making our data kind of unreliable and inconclusive.
Our next step for this project include further refining the oxygen testing techniques and reevaluating the optimal reference dye.
Thanks for taking the time to stop by our project. Hope you have a good day.